The next important transcription factors are transcription factor 2E and transcription factor 2H, that is TF2E and TF2H. TF2E, which consists of two subunits, binds next and has roles in the recruitment and regulation of TF2H. So the next transcription factor which has to come to the pre-initiation complex is TF2E, and it has a role in the recruitment of next transcription factor, which is TF2H. TF2H controls the ATP-dependent transition of the pre-initiation complex to the open complex. So now this pre-initiation complex will enter into the open complex, and this transition is ATP dependent. It is the largest and most complex of all the journal transcription factors because it has 10 subunits and a molecular mass comparable to that of the polymerase itself. So its molecular mass is about the same as that of polymerase. So here is the picture of whole pre-initiation complex, and in this picture you can see this is polymerase 2, and then this is INR region, this is TATA region along the DNA strand, and then we have different transcription factors such as this is TF2H, this TF2E, then TF2F, TF2B, this one is TBP which binds to TATA region, and this is mediator complex, this one is TF2A, this one is TF2D. So there are many transcription factors which are involved in the formation of pre-initiation complex. Within TF2H are two subunits that function as ATPases and another that is a protein kinase with roles in promoter melting and escape. Together with other factors, the ATPase subunits are also involved in the nucleotide excision repair. You know about the nucleotide excision repair. So these factors are also involved in this repair. How does TF2H mediate promoter melting? Because promoter has to melt. So how it is melted with the help of TF2H? It is now believed that a subunit of TF2H acts as an ATP-driven translocator of double-stranded DNA. So it is a translocator of double-stranded DNA, but this translocation is ATP-driven. This subunit binds to DNA downstream from polymerase and feeds double-stranded DNA. It binds downstream to polymerase and feeds double-stranded DNA because downstream DNA is double-stranded. So it feeds double-stranded DNA with a right-handed threading. So when it feeds with DNA, there is right-handed threading because DNA strands are left-handed coiled. So there is right-handed threading, which means it is opening up the two strands, and then it feeds these two strands into the cleft of the polymerase. This action drives the melting of DNA because the upstream promoter DNA is held in a fixed position by TF2D and the rest of the GTFs, journal transcription factors.